So the first speaker for this session is Helen Ginn. Helen completed her PhD at the University of Oxford. Her thesis focused on improving methods for data reduction for serial crystallography at synchrotrons and expels. In her current role as postdoctoral research associate at Diamond Light Source, she works on methods development for structural biology. Specifically, she has developed a new structural refinement program called Vagabond, which describes proteins through their bond network instead of atomic coordinates. She has also developed a clustering method for crystallographic datasets called Cluster4X, which she has recently used to contribute to the work mapping the antigenic surface for the receptor binding domain of the spike protein in SARS-CoV-2. And I believe she will be talking in more detail about that today. Helen, over to you.
Thank you. Let me just share my screen. Right, that should be visible, yeah.
Yes, it is.
Great. So yeah, thank you very much for the introduction, and thank you for inviting me to give a talk. I'm going to give a talk on Cluster4X, which has actually been developed in response to the coronavirus pandemic, and I'll give a little bit of detail about how it works and what you can use it for and how you can get it. So Cluster4X is was developed in response to a number of drug screens or fragment screens that were performed at a number of beamlines at the beginning of the pandemic, mainly against the coronavirus main protease. And these drug or fragment screens were focused on collecting data from a number of crystals of the main protease and then passing it into excellent software, Panda, written by Nick. What Panda needs is very isomorphous crystals, but unfortunately the main protease doesn't or didn't produce very isomorphous crystals at the beginning. So Cluster4X was actually written as a pre-sorting stage. So if we have a number of reflection lists, which I'm representing here with MTZ files, what Cluster4X was able to do was to actually resort them into sets which behaved in similar ways, so with similar crystal contents, and then passing those sets into Panda separately actually produced significantly more drug hits. But Cluster4X actually had uses that extended far beyond that, but I'll first explain how it worked for reflection lists. There are a number of clustering programs already available, so why did I decide to write another one? This is because I wanted it to be fast, really fast in real time, so that people could interact with it graphically. So it has a graphical user interface, and I wanted people to be able to really explore their data sets and get to grips with the pathologies within the data sets as well. So what Cluster4X does is to take a number of reflection lists and firstly merge them into a set of average reflections. We can then go back to the original data sets and scale the individual data sets in resolution bins in order to bring all of the data sets onto a sort of basic isomorphous similarity of the B factor. And after that, each of the individual reflections you can then subtract the average from each data set, and this produces a delta data set, which you can then look at as the deviation from the average for each data set. We can then go through every pair of data sets and calculate correlation coefficients to see which data sets behave in similar ways with respect to the average. So once you have such a set of correlation coefficients, you can then insert them into a matrix of looking at the pairs of reflection lists and deciding which ones behave in similar ways with happy faces and which ones behave in different ways with sad faces, and some which may behave similarly but not quite as much. And once you have such a matrix, you can then explore it in Cluster4X. So just to show you, you can download this quite easily on Linux if you have Snapcraft. Just type snap install cluster4x, and then you're able to view the correlation matrix, and it's quite interactive. So you have a number of options. You can also explore the data set and pick out clusters and recluster and recalculate averages and so on to really go into your data. But actually, that really helped with finding new hits in the data sets, but actually it was led to quite a lot of other stuff. So it led to, thank you for the introduction, led to antigenic mapping on the surface of the coronavirus receptor binding domain. So to give a little bit of information on that, the coronaviruses have a particular protein on its surface, the spike protein, which is responsible for getting into the human cell, which is not great. And the spike protein is one of the main targets of the antibodies which B cells produce in order to neutralize the virus. And what the Gavin Screeton lab did in the University of Oxford was to grab the blood of infected patients, and from those isolate the B cells from those patients which were reactive against the coronavirus, and from those isolate the antibodies which those B cells were programmed to produce. And once we have a set of those antibodies, we can then have a look at where the ones which bind to the spike protein, we can roughly bin them into where on the spike protein they bind. So the spike protein has two particular domains which are important, the receptor binding domain and the N-terminal domain. And once we have a number of isolated antibodies, we're then able to bin them, the Gavin Screeton lab, bin them into ones which bind to either the receptor binding domain, the N-terminal domain, or somewhere else on the spike protein. We're very interested in the ones which bind to the receptor binding domain, because these ones are the ones which are most, these ones are the most likely to actually give us neutralization capacity against the virus, where the antibody can prevent the virus directly from binding to a human cell. And so what Darming did in Oxford was to fix the receptor binding domain onto a biolayer infratometry layer, which is able to respond to the mass which has been bound to it. So if we were then to introduce a antibody which is able to bind to the receptor binding domain, then it will produce a 100% proportional signal boost for a single sort of antibody mass, one can think of it. And once we introduce a second antibody that's been isolated, it may or may not be able to bind at the same time. If it's able to bind at the same time, we'll see a 200% signal boost. So they don't compete, they're not binding to the same place in the receptor binding domain. Or we may choose an antibody which has to bind to the same place and is therefore unable to do so, in which case we'll just stick to a 100% antibody mass signal boost. And so once you have these values, you can look at every pair of antibodies that have been collected, and there were, and from this you can construct a competition matrix. So you can decide if, for example, there may be antibody pairs which were able to bind at the same time, which are represented by blue ticks here, or they're not able to bind at the same time, so they're competing, and those are shown with red crosses. So you can start to see the similarity here and why cluster4x was useful. The actual data was collected by Darming, who performed thousands of individual experiments on the 80 antibodies that were collected from the receptor binding domain, a really big piece of work. And this is the results. So here I've got the color scale here. So again, red is complete competition and 0 is no competition, but due to experimental variation, I extended the scale to yellow and black accordingly. and grey here are unrecorded values. And this is an unsorted matrix, so it's only using cluster4x and resorting the matrix to show that actually this, just by changing the rows and columns and recalculating the matrix, we can see that this roughly corresponds to 3 separate regions where the antibody wants to bind at a first glance on the receptor binding domain. But what we really want to know is where every single antibody actually binds on the receptor binding domain. And this is quite an undertaking, but during the course of this, this work, Helen, a different Helen, Ren and Darming in the lab were collecting structures of individual antibodies. So we knew the structures of some of the antibody receptor binding domain complexes, which I've just marked in yellow here. And when we have that information and we have the information of which antibodies compete with other ones, maybe we can begin to reconstruct a map of where every antibody collected binds on the surface of the receptor binding domain. And so for this, we take the receptor binding domain, which is actually more detailed at a molecular level. And in the rest of this talk, I will be showing antibody positions. The midpoint of the antibody binding is represented by a white blob. And actually, the surface of the receptor binding domain is a little bit too detailed. And so one of the first steps was to generate a mesh that gets shrink wrapped onto the surface of the receptor binding domain, and that gets rid of these crevices which actually antibodies aren't able to access, and introduce various local minima which would be nicer to smooth out. So this mesh really helps. And once you have a set of antibody positions, you can calculate a predicted competition matrix from any set. So for example, here, if you have 2 antibodies and you look at the distance between them and it's further apart than the diameter of one antibody, then you can say that they are able to bind at the same time. On the other hand, you may have some which are much closer together, in which case they're going to have a red value. So they do compete, they're not able to bind at the same time, and from that we can generate this predicted matrix. That's actually a sliding scale, so it goes smoothly from red to blue. And then we just want to jiggle around the antibody positions until that competition set looks like that of the data that Darming collected. And so once we do that, we can generate solutions. And if you do this multiple times, you actually end up with different kinds of solutions. So what you can see is that we end up with one kind of antibody set, which produces a really decent prediction of the antibody positions. And you can produce several different kinds where the antibodies are actually in completely different places. And what the hints are that you get these dropout values, like these 3 antibodies in the top group here, which means that this model is actually not that great. And the model below is actually closer to the real solution. And the reason this happens is because we're looking at a competition matrix, which is based on a distance matrix. Yeah, just highlighting those antibodies there. And so what you do is when you choose a set of antibody positions, they will produce exactly the same competition matrix as if you were to invert the set on the y-axis or the x-axis, or inversion of symmetry and so on. These will all produce very, very similar competition matrices. And the main ways that you are able to distinguish these is based on the surface of the receptor binding domain, which is a constraint. So they do have to bind to the surface, and the surface may allow that sensitivity to come through. Or you have a free set of structures where you've solved the structure, but you haven't included it in the data, kind of like an R preset, and you can use that to evaluate a given solution. And the solutions are also able to be pulled out because you can cluster on the antibody positions using cluster4x. And so the combination of these two methods allowed us to pull out the true binding map for the antibodies. And so when we have a look at that, we have here the real result that we can now create a much more detailed competition matrix from the data, so a predicted competition matrix with all of the values filled in, including the non-recorded values, and then cluster on that, and we can start to see that the clusters actually seem to fall into 5 main areas. We have the left shoulder, neck, and right shoulder, as we called them, and the right flank and the left flank. And the left flank is reasonably separate, and the other 4 produce a continuous epitope with subclusters. And if we have a look at the back of this receptor binding domain, you can see how the left shoulder, neck, and right shoulder all will compete roughly with each other, and the left flank is separate. The human receptor, the ACE2, actually binds across the left shoulder, neck, and right shoulder, which I've shown here. And if we color the antibodies by their ability to neutralize the virus and prevent infection, so here I've colored a good neutralizing virus in bright red and a bad neutralizer in blue, we can see that the bright red neutralizers actually bind in the same place as the ACE2 receptor. So that's a good cross-check as well to see that we've generated our map nicely. And so once we have this kind of method, we can then start to look at the variants. So one variant, the alpha variant, which was the first identified variant, also was known as the Kent variant in the UK and the UK variant outside of the UK. This shows, here I've plotted the depletion in the neutralization capacity of each antibody, which the Screeton lab measured, going from bright red, which is a pretty much annihilation of the antibody, to white, where the neutralization capacity wasn't actually affected. And we can see that for the alpha variant, there was minimal loss of antibodies. One antibody was completely destroyed, and the rest were reasonably well maintained. That's in contrast to the beta variant, B1351, where you got a significant loss of neutralization capacity in the region around 2 of the particular mutations in this variant, and there was some remaining capacity left, but it was significantly further away from the wild type. And preliminary data suggests that the Omicron map for this is just bright red, unfortunately. But this actually maps quite well onto another project, which I'm not going to go into the details of, but mapping the antigenic space of the coronavirus variants. So here we have, we're able to look at sera. When a patient has been infected with one variant, we can ask how well is it able to neutralize another variant, and the Screeton lab has again been generating neutralization titers for patient sera which have been infected with one variant, and checking against all of the other variants, and from that calculating a antigenic map where the distance between two variants represents how much more difficult the immune system has to cross-neutralize from one to the other. So, for example, here, if you've been infected with the alpha variant, then this is actually quite close to the generally considered wild type variants, and so the immune system is not evaded, whereas the beta variant is significantly further away. And that corresponds really nicely to the monoclonal data that we've been collecting as well. And they generally correspond, and we can see that the Omicron variant here has been blown out into orbit. So the immune evasion for Omicron is pretty high. And so, yeah, this has really helped our understanding of the coronavirus variants. And I would like to thank my lab, the Dave Stewart lab, and also the Gavin Screeton lab, and these are the main players, and we've really worked together to bring real understanding and overview to the coronavirus variant situation. So thank you very much, and I'd really be happy to take your questions.
Thank you, Helen. Sorry for this really nice talk. Always your animations are really nice to watch and very, very easy to understand as well. But we have quite a few questions. I thought the Q&A was disabled, but it seems to be very active.
Oh.
I will just start with that quickly. Vesna de Serrano, I'm not sure what you're talking about. Is it crystals generated with all these different antibodies, or i.e. cryo-EM?
I don't know exactly when that question.
This was actually at the beginning of your talk. I noticed it. I'll continue with he or she has several questions here. And relatedly, are you adding them to the spike protein all at the same time?
I would love to know exactly when these questions were asked, and then I'd or first doing some binding studies like with SPR or such. I think I covered that bit.
Yeah, they were quite early on in the talk, from judging from the, no, I don't know, I'm not sure. But yeah.
I think it was when you had the slides of the antibodies binding to the receptor binding domain. I think that's when these questions emerged.
Okay, so the tests are done on individual antibodies or pairs of antibodies, depending on whether we want to look at the binding of where a particular antibody is roughly, or the pairs of antibodies, which to understand the competition between two pairs.
Okay. Next question. Could the distance between a new variant and others, as shown on the last slide, be used as a sort of infectivity measure based on how it may invade the immune system?
Yes.
Sorry.
So in fact, it can't really tell you whether it's more infectious or not, but it can tell you whether it's going to evade the immune system more easily. So, partially, like, it answers half of that question in terms of evasion of the immune system, but it cannot tell you how easily it infects a cell without any immune system response involved.
Okay. Next one, as usual, awesome handmade graphics, wonderful talk. Sorry, I missed this point. Does matrix 4X work with data that have different unit cells? Also, I see it nicely working on antibodies. Do you think this method can be applied to a small molecule drug screening?
So cluster 4X, yes, it does work with different unit cells. It will generally cluster on the differences between the unit cells and correlate with unit cell differences. It's a bit more sensitive than clustering on the unit cells themselves, and those will generally come out in the wash. So yes, you can use it with actually any data, not even related to structural biology. And particularly, yes, unit cells, it will end up clustering on the differences because the amplitudes are correlated with unit cell changes. Yeah, the method was actually originally developed for a small molecule drug screen. So that was the origin, and it works really well there.
Thank you. Next question. If the virus variants have mutations on the RBD, very close or actually where the RBD binds to the ACE receptor, should it be good in the sense that then the RBD will not bind the ACE receptor? So that the fact that the antibodies don't bind the mutant RBD anymore is not as bad as one might think, maybe?
Yeah, so I totally get the question. I think if the virus were in equilibrium with the humanity for a very long time, then yes, it would be good. But at the moment, because it has only just escaped from the animal kingdom, there are still a number of mutations which have been explored through the variants and Omicron, where the binding is actually improving to ACE2 at the same time as evading the immune system. At some point, that will lead to a minimum where it can't bind the ACE2 any higher, or it's no longer the limiting factor in the infection process, after which situation will apply where any evasion from the immune system has to be accompanied with a reduction in the binding capacity to ACE2. Omicron may be getting towards that minimum, so there's been really good studies by Gideon Schreiber and his group, which have actually generated through mutagenesis a very tightly binding receptor binding domain, which until Omicron appeared, most of those mutations had not yet been sampled in the general genomes, but now with Omicron, most of the major players in that receptor binding domain that he created and his lab created were, most of those have now been found through Omicron. So maybe we're starting to get to that minimum, the maximum binding potential of ACE2, and then this situation will apply.
Thank you. There's more in the Q&A, but I'd like to go to Slack as well. Melanie, are the antibodies pre-validated as RBD binders? Is neutralization predicted or experimentally measured?
I missed the first question. Neutralization is experimentally measured. What was the first part? Sorry.
The first one is, are the antibodies pre-validated as RBD binders?
Yeah, so there was a first step of finding antibodies which were isolated from B cells which were reactive against coronavirus, and then a spike screen, so it had to be reactive against the spike. And then out of about 400 antibodies, 80 of those were pre-screened to be binding to the receptor binding domain before they went into this competition matrix.
Another question from Elliot. Cluster 4X speedily and graphically clusters pairwise comparisons. What are the differences in the pre-processing of reflections before clustering versus the processing of the antibody competition matrix data?
So the antibody competition matrix data was, was generating a competition value based on this 200% signal boost, 100% signal situation. So that was just inverted, so that a competition matrix gave a value of 1, and no competition gave a value of 0, and that was actually extended because of experimental variation, like some antibodies falling off, which meant that it went over 100% or below 0% competition. That's all that was done in terms of pre-processing of the data there, so pretty much nothing.
Lots of comments on how nice the talk was. Amazing talk. Just quickly picking up some more questions. Could you speculate, for the mapping to work, what would be the minimum ratio of known binding sites versus unknown?
In terms of known, experimentally determined, or unknown. So all of the antibodies which were given a predicted location. There were 1 or 2 which didn't behave very well and couldn't really decide where to turn up on the receptor binding domain. That could also be due to problems with the experiment. Some antibodies may have caused the receptor binding domain to fall off the instrument. That there was some indication that was happening as well. So some of the antibodies didn't behave very well, and then in the end, maybe 1, 2, or 3 were excluded because they weren't behaving nicely, out of 80 also.
Thank you, Helen. There are quite a few more questions. I'm not sure whether we should, in the interest of time, carry on.
I saw, so methods that were used, there was a combination of core-EM structures and crystal structures by the Stewart lab.
There is quite a few more in Slack. Have or could any of these results be validated by SACS rather than MX?
I haven't really thought about that. I guess theoretically, yes, that would be a way of, sorry, the MX was not to determine the competition values. The SACS could theoretically be used to determine competition values, I think. But the MX was used to determine some of the structures.
Can this methodology help predict ADE, antibody-directed enhancement, as seen in dengue and Zika viruses?
The antibody mapping itself, we didn't see any. So that's more from the neutralization capacity. So if adding an antibody were to make it more easy to for the coronavirus to infect the cells, then that's how you'd be able to identify antibody-dependent enhancement. There really hasn't been that much of this seen in coronavirus, thankfully. It was a big worry at the beginning, but generally we haven't seen too much of that happening. I think maybe some labs have reported, but it's not a major player, thankfully.
Thank you. I'm just wondering, there are a few more questions. Are you happy to have a look at Slack yourself, Helen?
I can answer them separately.
Just because I think we've
other people need to share the time. So I think that's good for me.
Thank you, Helen.
Thank you.